For coming to a Scientific Computing and Imaging Institute Distinguished Lecture. We are very pleased to have Terry Sejnowski here today as our distinguished lecturer. Terry heads the Computational Neurobiology Laboratory at the Salk Institute, and he has a large number of awards and honors. He's one of the few people who's in all of the national academies of engineering, of science, and of medicine. Other fellows and associations he's belonged to. Terry started out in physics, like I did. So as we were exchanging stories this morning, and got his PhD in physics from Princeton, but then changed over to biology as a postdoc. And I was trying to think the first time that I had heard his name, and it was back in the late 90s after he and Bell invented the independent component analysis algorithm. And soon thereafter, I actually wrote a paper with a couple of my students on using the independent component analysis to solve a problem in electroencephalography of source localization. And he has used that algorithm for many, many different applications and doing many other very interesting work in computational neurobiology. So he's going to speak on space-time computing and dendritic spines today. Please join me in welcoming Terry Sejnowski to the University of Utah.
Thank you. Well, it's just wonderful to be here. I've been to Salt Lake City many times for meetings. So early Snowbird meetings and neural networks, that was back in the 80s, and then the cosine meetings that have been going on for the last 20 years. But this is my first visit here to the university. And I'm particularly pleased to be here giving a talk in ski because it's an interdisciplinary institute, and my work over the years has been very highly interdisciplinary. And I really think that that is the future. If you're a young student, don't put yourself in a silo and consign yourself to be a narrow, very constrained in terms of your interests or your tools that you're going to be using. But by working across fields, and that's really where the really big problems are today, like the brain. It's one of the biggest problems. And there's no way that any single discipline is going to crack that problem. And this is actually the goal of the BRAIN initiative you probably heard about. In April 2nd, 2013, President Obama announced that the BRAIN initiative was going to be his signature science project. And these are only announced once every decade or two. You know about Kennedy's putting a man on the moon, the Human Genome Project. This is going to go on for 10 years. And the goal, interestingly, is the I and the N. It's an acronym, by the way. BRAIN is an acronym. Innovative Neurotechnology, IN. And what that means is that we need neuroscientists to work closely with engineers, physicists, mathematicians, who have the tools and techniques that can help us make progress. And it's been spectacularly successful. I've been talking with a few people who have BRAIN initiative grants, and it's working exactly the way that we had hoped. Today's lecture is going to be focused on the dendritic spine. This is a very peculiar structure that is found across all animal kingdoms. These are integrative synaptic units. One of the most complex macromolecular complexes in the body is the postsynaptic density that contains 400 different types of proteins. And in fact, if you look at all the genes that go into assembling a synapse, it's about 2,000 genes, which we're talking about 10% of the whole genome, has a role to play in communication between neurons. Now, if you look at the different levels that you can study the brain at, all the way from the molecular level at the very bottom here, all the way to the central nervous system where behavior is generated, there's 11 orders of spatial magnitude. It's an enormously multi-scale problem that we're dealing with. And there's interesting structure at each one of these levels. And within the last decade, with new techniques being developed for working out connectivity, connectomics, as it's now called, has developed techniques, for example, for using diffusion tensor imaging for creating long-range connections at the systems level. And then down here at the network level for microconnectomics, for being able to work out local circuitry that has to do with the high degree of connectivity, say within a column of cortex with about 200,000 neurons. There's roughly, a billion synapses, right? And so working out just the local connectivity in a little cube of the cortex is itself a major goal for the connectomic field. Now, what I'm going to be telling you about today is going to start at that level, microconnectomics. This is an example of a dendrite, the pyramidal cell in the hippocampus, and here is a spine, a neck, and here's the head. And in blue, here is an axonal terminal. This is an axon that is going to communicate with the postsynaptic spine. This is a freeze fracture picture, and by the way, these colors are not there in the real tissue. But we're doing that so that you can see it, because otherwise it's a little bit difficult to interpret. It's like the moon, surface of the moon. But what we're interested in doing is to extend connectomics down to the nano level. That is to say, to really be very precise about the sizes and shapes of each one of these structures, and then to know exactly where the molecules are located, the receptors on the postsynaptic side, the release mechanisms, and all of the machinery inside. We want to be able to understand how all of that machinery is used under different conditions to signal an event. And then what happens over the history is that the size and the strength of the synapse changes. And that is how learning and memory, we think, is instantiated in the brain, is the ability of patterns of activity to activate long-term potentiation, increasing the strength, or long-term depression. Now, this is a very beautiful drawing from Ramon y Cajal, the great Spanish anatomist, who synthesized a circuit diagram for the hippocampus. And what you're seeing here is actually not a photograph. He actually looked through Golgi slides during the day, and then at night he would, from memory, right, what he saw was a circuit. And so remarkably, I mean, he was accurate, extremely accurate, and very prescient in terms of understanding the mechanisms. So here we have inputs coming in to the input layer called the dentate gyrus. And then from there, they go to an area here called CA3, where there's recurrent connections. And then from here, the axons go through a Schaffer collateral to CA1. And in this pyramidal cells, you can see these little spines coming off. He saw the spines, even though they're very small. They're less than a micron, but he saw them in Golgi. Is the best studied synaptic plasticity in that synapse between the Schaffer collateral and the dendrites of the CA1 pyramidal cells. By the way, the reason I know that the information is flowing in this direction is that I'm just following the arrows. Again, the arrows were not there in the tissue. And he just, literally, he deduced this from what he thought was how the circuits are actually organized in terms of the neuron doctrine of the information coming down the dendrites and going out the axon. So this is really amazing. Now, what we're going to do, I'm going to be telling you today about a project that was started with Kristen Harris over 10 years ago. And two stories. And it's all about what happens in that little box. That little box is 5 microns on a side. So this is a tiny little piece. But we want to reconstruct it completely. Every single synapse, dendrite, axon, and astrocyte, glial cells. We often forget about them, but they play a very crucial role in the function of that little piece of neuropil. So here's what a cross-section looks like, and we're going to do 3D reconstruction of EM, transmission electron microscopy, 1 micron across. This is a dendrite here. You can tell that this is a presynaptic terminal because it's loaded with these vesicles, and these vesicles are released one at a time. And this is a cross-section of a spine head. You can't see here where it goes because it's just, you have to follow it through cross-sections in order to see how it connects up to the dendrite. Okay, so we did the reconstruction using computational geometry, and here is the problem. The problem is within plane, the resolution is 4 nanometers, which is really good. But between planes, it's 40 nanometers, a factor of 10 worse. And being able to connect a particular location here with a particular location here is ambiguous. And so, in fact, this is what we started out by segmenting, and that was what Kristen did for us. And then we took this and then reconstructed the third dimension. The correspondence problem has to be solved. There are, we do have some algorithms from computational geometry that is able to do the bulk of it, but it turns out that it wasn't designed for this kind of tissue, and there are particular geometric problems with it that have to be handled individually by hand. So it was curated by Justin Kinney, who was a graduate student at the time. And at the end of the day, I should say at the end of 3 years, we arrived at this Chinese puzzle. We've color-coded the different pieces here, and I'll be telling you about that in a minute, except to say that it's a densely packed piece of tissue, and it's very difficult to understand the geometry. So I'm going to unpack it for you. I'm going to play a movie, take it apart, put it back together. And so that way, at the end, you're going to see the intricate, three-dimensional, entangled pieces of a little piece of neuropil, but it will be, we'll see at the end that it will reveal very interesting conclusions. So this is the central dendrite. There are 75 synapses, and each one of them is on one of these spines. This is a little branch coming off. Here comes the axon. So each one of these axons, 75 axons, is going to contact one of those postsynaptic densities in red. That is where the receptors are located. Now there is only a single piece of an astrocyte that is responsible for enwrapping the synapses in this particular volume. These things are 30 microns across, so this is a 5 by 5 by 5 cubic micron chunk of a single astrocyte. But it fits in between the pieces here like the fingers in a glove. So now what we're going to do is to dive down into that central dendrite and see what's on the inside. You can reconstruct the endoplasmic reticulum, mitochondria. So this mustard-looking snake here is the mitochondria. They're very long and thin. And these are fragments of the endoplasmic reticulum, which should be continuous, but was not in our reconstruction. We need higher resolution in order to be able to do that. But now what we'll do is go through one of the spine necks. It's like a tunnel, about a micron long. And you can see the light at the end of the tunnel. It's the postsynaptic density right there in red. And that is the business end that has the receptors that will transduce the neurotransmitters into an electrical signal. Looking back, you can see now the presynaptic terminal here in gray. And here's another synapse. Now, according to the textbook, this should be wrapped around with an astrocyte. The reason is that the astrocytes take up the glutamate neurotransmitter when it's released. But as you can see, it has to travel some distance in order to get to the astrocyte where it binds to the transporters. This one has one close. We're going to see a classical textbook case in a minute. By the way, almost everything you read in the textbook is wrong. So here's a nice wrap. What you see in the textbook is kind of ideal circumstances. So this one is nicely wrapped around, capturing all the glutamate as it comes. It also doesn't spill out. For most synapses, it does spill out. The other thing is that they'll tell you that on average there's 1 synapse per micron, so there should be 5 synapses on that dendrite, or there are 75, which means that it's like 15 per micron. Okay, so we're going to reconstruct the whole thing now. This is just a little piece of it. So here are all the rest of the dendrites. These are really fragments of dendrites, but they're all passing through, forming 400 synapses onto the axons. Here comes the 150 axons coming through, or rather more like 400. So 150 dendrites, roughly 400 axons. It's the 3D structure is really incredibly complex, and once you get to know it, very intricate. But it actually is revealing. I mean, it is hitting inner simplicity. So this is the hole through which the central dendrite goes. This is just the axons and the rest of the elements. We've taken away the dendrites. Okay, so that gives you... There was Blue Danube, by the way. Okay, so the first thing, we wanted to be as accurate as we could possibly be. So the first thing we did was to take the reconstruction and to blow it up. And that's because during the processing for electron microscopy, it's dehydrated and the extracellular space shrinks. And we know that because the extracellular space has been measured in vivo. Its volume fraction is 20% in this piece of tissue. And it gets compressed down to a few percent here in the reconstruction. So the first thing we have to do is to expand that. Justin did that by mechanically putting springs in between and letting it expand to 20%. And one of the things that he immediately hit on was the fact that it's not uniform. The extracellular space varies with the location. So if you're, for example, between two surfaces, flat planes, as shown here, then it turns out that the spacing here tends to be on the order of 10 to 20 nanometers, which is what you read in the textbooks. The textbooks, again, tell you what is typical, but it's not true everywhere. For example, if you are at a cross-section here where there are 3 elements, then you can't have, the curvature is limited. And so therefore, the spacing here tends to be much bigger, on the order of roughly 40 to 80 nanometers. Now, this is a cross-section. And so if you actually extend this in 3D, then this becomes a sheet and this becomes a tunnel. And that's illustrated here. These are the crossings with 3 different structures coming together, the tunnels. And here is the sheets in between in red. Now, there's one additional parameter, and that has to do with the diffusion. You can measure the diffusion constant, and you need to get that accurate too. And so it turns out there's one more degree of freedom, which has to do with the degree of what we call lacunarity. That's a degree to which you have some regions in which there's actually a big pool of extracellular space. You can see this one, the sort of the little pools between the structures are much bigger than here. We can vary that parameter, and in order to adjust it to the diffusion constant, because the more space you have to diffuse, the faster the little molecules can travel, and that's been measured. And interestingly, since we did this, which was about, I think, roughly 8 years ago, there have been new techniques that have been able to come up with ways with fast freezing and cryomicroscopy to be able to reconstruct this more accurately. And it turns out that this one here with the big holes in it are probably closer to ground truth. And this is the one we actually hit upon. For other reasons, we thought that this was probably closer to the actual tissue. So this is great because now we can use it for simulations. And the simulations, well, first I want to show you an image that you've never seen before. In fact, nobody's ever seen this before. And imagine, instead of thinking of the extracellular space as the outside, imagine it's the inside of some very complex three-dimensional structure, right? So we get rid of all the cells, we get rid of all of the dendrites, and we just say what's left at the end of the day. And that is topologically incredibly interesting. And here it is. So what Justin has done here is put a blue dot when you're in a sheet, so you're 10 to 20 nanometers apart here at a surface, and yellow is when you're above 20 nanometers. And you can see the tunnels, and it's amazing. And it explains something in literature which was a paradox. Because if it was the case that all of the distances between the different parts of the cell, the different parts of the neuron in Neuropil was 10 to 20, how could you explain the fact that quantum dots, which are 80 nanometers, could diffuse through the tissue, right? That doesn't make any sense. And now we know it's these tunnels. It's a kind of superhighways running through the Neuropil that large molecules can travel through. But it's really a beautiful structure. I mean, just mathematically, just being able to look at it and analyze it is really quite interesting. Okay, so this is a prelude to the fact that we want to be able to simulate the physiology. That is to say, what happens when the action potential comes down the axon and releases transmitter and diffuses over to the postsynaptic side where it binds to the receptors. Each one of those has been studied in great detail by anatomists, physiologists, biochemists. And so what we wanted to do here is to put together all of those components, the anatomy, the physiology, and the biochemistry, which live in different silos. But by putting them together into the simulation, we can then come up with predictions and conclusions that you could not have come to by just looking at one of those data sets. I'm not going to go through all the details. Obviously, there's a lot of other components here, voltage-dependent calcium channels. There are calcium binding proteins that are very important for regulating free calcium inside the spine. And then there are calcium pumps that allow you to clear the calcium after they come in, primarily through these voltage-dependent calcium channels, but also through the NMDA receptors, which we'll be hearing more about later. We can also take advantage of the fact that a lot is known about the kinetics of these ion channels. There's a stochastic process. Markov models have been developed, very accurate ones that we use. So here's the snapshots from a particular release. So that's a vesicle that's just released the transmitter, and in the background you can see the actual receptors. The AMPA receptors are in gray, and there's typically on the order of 20 to 100, depending on the size of the spine head, and maybe 10 to 20 NMDA receptors. Now, one of the things I want to point out to you here, which is really critical, is how small the numbers are here. We're not talking about Avogadro's number, right? We're talking about handful. And that means that Monte Carlo techniques are very efficient. And Monte Carlo techniques developed by the physicists back in World War II, and what you do is you follow diffusion by a random walk. And our time steps are 1 microsecond, so you decide randomly where you're going to go in a random direction, and you take a distance step which follows the diffusion law. And here is how very fast these glutamate molecules, the ones in blue here, how fast they diffuse. In the first tens of microseconds, they're already streaming out from the cleft between the pre and post. And by 100 milliseconds, they're completely gone. And what's left are the binding of the glutamate to the receptors. And what you see here, and this is 8 milliseconds later, is the NMDA receptor opening and closing as it chatters and lets calcium ions come through. Now, the background level of calcium ions, free calcium, is extremely low. In a spine head here, which has this is not typical, but it's on the order of a few hundred nanometers, with 200 nanomolar background levels of calcium, there's on average 7 plus or minus 3 free calcium ions. Again, we're talking about small numbers. And what this also illustrates is that the traditional way that you go about solving this problem is using finite element methods. You'd put little volume elements in here, and you assign a concentration to each, and you figure out where the concentrations are going up and down. The problem here is that these molecules are bouncing around, and in most of the volume elements, there's no calcium. And then sometimes there's 1, maybe there's 2. It's a very finite problem in terms of the numbers. And using the traditional techniques, all you get out is a mean, which isn't even meaning. It's not even have meaning. It's meaningless, because what's an average of 0.01 per cubic nanometer. It doesn't mean anything. But following each molecule, we know where they are, we know what they're going to interact with, we know where they're going, and so we really, with this technique, we can really be very, very precise. Okay, so I'm going to show you another simulation, which took literally about 10 years to reconstruct. We have very detailed models of both the pre and post, and I haven't told you yet about the presynaptic side, but we're going to show you a simulation where you can actually see the process whereby the vesicle is released. So here's a cartoon of what you find on the presynaptic side. First of all, you find docked vesicles, and the red is a lot of machinery called the snare complex with calcium sensors. Calcium comes in through these clustered receptors. There are only a few of them. Typically, actually, this is misleading. There could be, just in this neighborhood, maybe 5 or so, but at most. The calcium then binds, and then at most 1 vesicle is released. The calcium here, as you can see in yellow, binds to the calcium binding proteins. So the free calcium goes very rapidly down, and then eventually is released and goes to the pumps, the calcium pumps that clear out the calcium and returns to normal, but that takes much longer. So that's the machinery on the presynaptic side. Here's the real complexity. And you can see the endoplasmic reticulum. It turns out the calcium comes in. It can also bind to ryanodine receptors, and it can also, the calcium can be pumped into the endoplasmic reticulum, and sometimes actually the IP3 receptors can come out. There's a lot of dynamics that occurs inside the axon, in fact, which is very important. And there's mitochondria, of course, which is the power pack, which is absolutely essential. So I'm just summarizing here very, very rapidly a lot of the machinery that I'm not going to tell you about. But rather, let me just play the simulation. I think, if a picture is worth 1,000 words, then the simulation is probably worth a few million. Okay, and so there's a logarithmic time scale here. So this is now milliseconds. And here's the synapse that we're going to be zooming in on. So here's the axon in green. And there's going to be an action potential coming down in a millisecond or two. But I just want to get, this is the ground state. You can see that free calcium is very, very sparse on the presynaptic side. So here are the vesicles. Here's the active zone where they're docked. These are the receptors in the postsynaptic density. And so we're about to see, here are the voltage-dependent calcium channels, and we're about to see the entry. So here is the first burst as these calcium channels open and close, and now is a boatload of calcium coming in, and it's about to release a vesicle. Right about now. There we go. That was the glutamate, about 2,000 glutamate molecules, these little fleas, and instantly it's cleared, starts binding to the astrocytes, and now the receptors are being occupied, and gradually over time, we're up to about 8 milliseconds now, the NMDA receptors are just beginning to open. AMPA receptors are very fast. They give you a fast EPSP, which is on the order of 1 or 2 milliseconds. But now the calcium is coming in through the NMDA receptors that are chattering. These NMDA receptors are voltage dependent as well as glutamate dependent. Now what you begin to see is that the calcium binds to calmodulin, and the calmodulin then binds to a very large molecule called CAM kinase 2. It's an important enzyme in the pathway toward LTP. And in fact, it's through a very high degree of activation of CAM kinase 2 that you do activate it's a kinase, activate other molecules downstream. Now, what you just saw there was a second release. That's an asynchronous release. There's a certain probability after activation through the release of transmitter that another one will happen within 5-10 milliseconds or even later. But in this case, now you're beginning to see a lot more activation of CAM kinase 2. So this gives you a feeling for the dynamics on different time scales, on the microsecond time scale, on the millisecond time scale, and this NMDA receptors can continue to open and close over 100 milliseconds. So this is several orders of magnitude. Okay, now, again, you've seen an end-to-end simulation, everything from the beginning action potential all the way to the effector, the CAM kinase 2. So this is a big stretch in terms of all the steps that you have to put in. I just want to give you a flavor for the pain that we went through with Mary Kennedy, who's a biochemist at Caltech, who studies chem kinase too, by the way, to actually pin it down. So one of the things that you need to know is something about the buffering capacity of the calcium-binding proteins. And the way you find out about that is by putting in calcium indicators with different concentrations that have a different buffering capacity, and you then measure the peak of the calcium coming in, concentration of calcium at peak, and the decay time in milliseconds. And this is done by Sabatini, who did it for both the spines and the shaft, and his data are shown here in blue. And so there are free parameters, as you can imagine, having to do with the time constants of the buffering proteins and the concentration of buffering proteins, the binding proteins. And so we adjusted those to try to match as well as we could to the actual experimental evidence. And that was very important because you have to calibrate things. So we think that we're pretty much in the same ballpark that we think the cell might be. Now, here is what you can do with the model that you can't do with experiment, just to give you, again, a taste of the consequences of having gone through this exercise. So first of all, we're going to be doing the simulations of two different spines. Here's a small spine, and here's a very big one. There's 2 orders of magnitude in terms of the differences in the size of the spines. And what we're doing here is comparing, we put into our model flow 4, which is a calcium indicator. We know the on and off rates, so we can do that very accurately. And what we do is put it in and then measure the fluorescence and then use the very same algorithm that the physiologist uses to convert the fluorescence to calcium concentration. If you do that, you get this red curve for the small one, and here's another red curve for the big one. Now, you might think that, oh, gee, these are very similar, but look at the y-axis here. You can see that the peak here is on the order of, it's on the order of 2 micromolar, whereas down here it's about 0.2. And that's because there's a much more volume here. And so the concentration is much less for the same number of molecules coming in. So that's known. I mean, that's something that we appreciated without having the model. But at least we've confirmed it. And now what we're going to do is to actually, we know how many free calcium there is. We're actually following every one of them. So the question is, what is the real concentration, right? And in green, here is the real concentration. So you can see that it's 4 times higher. In fact, the entire transient at the beginning is basically filtered out by the fluorescence that is coming out of, which is on rate is slow and so forth. And you get both the peak wrong and you get the decay wrong. Okay, well, so that means that the numbers that you see for calcium concentrations are not to be believed in people's papers. They can be off by factors of 2 to 10, depending on what kind of a spine you have. And then it gets worse, because it turns out that the calcium indicator itself is a buffer. So what we can do is, after we calibrated it, we can take the calcium indicator out and see what would have been under normal conditions without the intervention, right? You know, your measuring instrument often affects the actual measurement. And so here it is in blue. You can see it's significantly higher and it has a different decay. So this is what we can do. We can deconvolve the experimental measurements. We can actually tell you what the real ground truth is once we calibrated it. And now what we want to do is to use it to answer a question that has been an extremely important one, having to do with the conditions under which the synaptic spine can change its strength. And it goes back to experiments that were done in the 90s, initially by Henry Markham and Bert Sockman in the cerebral cortex, but this is from Mu-Ming Pu's lab on the hippocampus, where he took a lot more data points, so it's a lot more easy to explain. So it's a slice preparation. You have 2 pyramidal cells in the hippocampus, CA1, very one we're studying. And what you can do is independently activate the spikes in one or the other. And then you could pair the pre and the post. So here's a case where the presynaptic, the red one here, was stimulated with a time delayed by delta t. And then the postsynaptic was stimulated, and now we're looking at the postsynaptic spike. This is the EPSP, excitatory postsynaptic potential. And now we pair this at between 10 and 40 hertz. It turns out as long as you're above 10 hertz, you get the same results. And then you compare the strength of the synapse before and after, and within minutes, it changes. So you can see that if you do the pairing with a time difference of about 10 to 20 milliseconds, you can double the strength of the synapse. That's long-term potentiation. What if you reverse? Instead of pre before post, you do post before pre, and then pair that at above 10 hertz. And you can see now that it is a window here of, again, 10 to 20 milliseconds, where the pairing reduces the strength of the synapse by a factor of 2. So this is really important because previous to this, the best way that they used to stimulate LTP was by 100 hertz for a second, which is not physiological. You never get that rate for that long under normal physiological conditions. But you can see here, at normal rates, 10 to 40 hertz, and with relatively a few pairings, you can actually move the strength of the synapse around, but it all depends on the relative timing. Plus or minus 20 milliseconds is a very, very small change in terms of the relative timing, and you can see right at the 0, it's a knife edge. You're a little bit to the right, and you're here, and you're potentially a little bit to the left. So there's got to be a millisecond machine there keeping track of what the relative timing is. And so it's hard to even imagine biochemistry being up to that, right? To the strength, to the timing of the electrical signals. And this is especially true. Timing is really important for these ion channels. And so here are the best models we have for the AMPA receptor, the NMDA receptor, and other receptors, and voltage-dependent calcium currents. And you can see that there's a lot of closed states here that glutamate is coming on and off. We know that, for example, to open the channel, you have to bind two glutamates, and so it's a very complex process. NMDA receptor is actually even more complex than this. We're actually working on the second generation now because it's now known that it's a tetramer. So we really have a much more complex machine there. And there's a magnesium block, which means that in order to be able to get current, you have to depolarize. And if not, then very little current goes through. But when you depolarize and when glutamate binds, you can get calcium current, and that's what you were seeing. Okay, so here's the simulation. Here's the answer. How do you depolarize the postsynaptic side? Well, it turns out there are active currents that causes a back-propagating action potential. So you can actually get a very strong depolarization occurring in the dendritic shaft that I showed you there in the pyramidal cell, way far away from the soma. And if you just look at a single back-propagating action potential, it will open to voltage-dependent calcium currents, and you can see two channels that are open here. There's only typically on the postsynaptic side 2 or 3 channels, so it's very sparse. And you can see here that you get a significant amount of calcium coming in, and I'm showing you here with and without the buffering, but look at the gray because that's probably the realistic condition. So you get a few micromolar coming in very transiently. Now, what about the EPSP? So let's just do the EPSP by itself. So we have the action potential with the glutamate coming out. It binds to the NMDA receptors. And you may get a flicker here and there, and there's almost no calcium coming out. Why? It's because it won't open unless it's depolarized. So let's do that. Let's put the back-propagating action potential at the same time as the EPSP. And now we get a boatload of NMDA receptors and calcium coming in. And now you can see it rises to 10 micromolar, which is in the range to trigger LTP, and it stays around for a long time, which is also necessary in order to get LTP. So this is really telling us that it's the relative timing between these two signals. If they have to be there at exactly the right time in order to get this boatload of calcium coming in. Interestingly, if you do it the opposite order, I'm not showing you, you get calcium coming in, but much, much less, and that's the condition that we think leads to LTD. So there's a way to be able to predict. Now the reason why we really put a lot of effort in making this model as accurate as possible, and the reason is that in the real world, this axon is carrying a pattern of spikes, not just pairs. And so we really have the machinery now, so we can tell you with any pattern, with any history is going to give you, that you can't even predict from just the experiments that are out there. But we've calibrated it, so we think we'll be able to do that. Now, the other thing we can do is keep track of every calcium ion. And the question you might ask is, what happens after the calcium comes in? And there are a lot of ways it can get out. It can go out through the neck. Calcium pumps can pump it out through the spine head. Calcium binding proteins for a while will hold on to it. And you can see here in the normal condition with the pumps at the density that we know exists, that the free calcium is really just a very, very, very few molecules that are available for being able to activate different molecules. So what this is telling us is that the bulk goes out through the pumps and the rest through the neck, and that's it. But if we reduce the calcium pump density, then most of them are going out through the neck, and the calcium binding proteins are actually going out through the, as it turns out, carrying away the calcium. So this is, these are two extremes, and now we can actually make predictions for different disorders where the calcium pumps are either disabled, or there's several different genes that have calcium pumps, and there are also calcium pumps on the endoplasmic reticulum. So by studying, by disabling one or another of those, we can make predictions for what the calcium handling is likely to be and the impact that's going to have on things like synaptic plasticity. So this can be used for making predictions. Okay, I'm going to switch now in the last 10 minutes of the talk and tell you about something that was out of the blue. It's not something we planned, and this is one of those serendipitous things that really makes science exciting. So here's an observation that was made quite a long time ago by Chuck Stevens. It had to do with the correlation between the anatomical measurements. If you look at the strength of a postsynaptic potential, the strength of the synapse, right? How big of a EPSP are you going to get? It turns out to be proportional to the volume of the spine head. It's also proportional to the size of the postsynaptic density, which is proportional to the number of receptors. And it's also proportional to the size of the presynaptic active zone and the number of docked vesicles. Those are all regulated, very highly regulated. So if you have a big spine, you have a big presynaptic release site, you have a lot of vesicles lined up, and so all of that has to be communicated back and forth. So here was a reconstruction of every single spine on that apical dendrite you saw at the very beginning. And what this illustrates is a fantastic variety of not just sizes, but look at the spine neck length. Here's a big one, here's a small one. Spine neck diameter, here's a big one, and there's a tiny one. So these parameters are all over the place. And you might think that, well, geez, nature can't get it exactly right. This is biology. Well, we'll see. So looking at the distribution of spine head volumes, this is femtoliters. You can see that there's a peak down here. Most of them are small, but there's a very long tail. And these are the really big ones. 2 orders of magnitude. Now, Justin, who had been looking at these reconstructions for years, once called me to his monitor and said, Terry, look at this, take a look at this. Right? According to the textbook, when an axon goes through perpendicularly, it only makes one synapse. But look at this axon. It's making 2 synapses on the same dendrite. Isn't that interesting? I said, well, that's probably a statistical thing. It probably happens occasionally, but may not mean very much because, it's rare. And so he started collecting more and more of these pairs. And he also noticed that, gee, isn't it interesting that this volume of this head is about the same as the volume here? And so here are the pairs that he picked out from that. These are pairs of spine heads on the same dendrite activated by the same axon. So there are 10 pairs. And then there's one odd one here, which is a triple. And it's an anomaly, and I won't go into the details except to say that we excluded it. So let's plot the volume of one against the other. So here it is on the log scale, head volume 1 versus head volume 2. And you can see that it falls remarkably close to a straight line. In fact, the variance is 0.92 of the jitter, and the coefficient of variation, average coefficient of variation, is 0.083. That is amazing. And why is it amazing? So here is the reason. Synaptic plasticity is a function of two different variables, the presynaptic release, postsynaptic membrane potential, right? Now, we don't really know what the rule is for synaptic plasticity, but it's a function of those two things. These pairs of synapses that we're seeing here received exactly the same history of presynaptic input and postsynaptic membrane potential because they're right next to each other. So that means that whatever the plasticity role, they got the same inputs. And now we can ask how precisely is the rule that produces synaptic strength. And since the head volume is a proxy, and the fact that we have seen that they're very accurate means we can put a limit on the precision of the rule for synaptic plasticity. And the reason that's important is that we have speculated for decades on how many bits of information you could store in a synapse. Now, from that distribution I showed you, a lot of people concluded that there were one bit of information. It's either big or it's small, right? That's kind of the crudest thing that you can imagine. But then people will say, well, wait a second now, there's a couple of gradations out there. Maybe there's 2 bits of information. Okay, well, we're in a position here to actually put a real quantitative estimate on that number. First, just to convince you that this is real, we looked at the distribution of axon-coupled spines on different dendrites just to see if it had something to do with the axon itself, and you can see it's not at all well correlated. Now, here's what we did. This is a little bit complicated, and not everybody is going to follow this because it really depends on knowing something about signal detection theory. And it has to do with the fact that if you're drawing from 2 distributions, what's the probability that you're going to be able to accurately know whether your synaptic strength is here or here, right? You're given some measurement. It could be anywhere in between across these two. And you have to assign it to either this strength or that strength, right? That's signal detection theory. Well, if it turns out that with CVs of 0.084 and a detection threshold of 69%, being able to accurately assign it to one or another of these bins, you can fit in about 26 different strengths, and that corresponds to 4.7 bits. So this is really pretty good machinery here. We're talking about storing, roughly 5 bits of information, and for computer scientists, that's doing pretty good. I mean, you have 10 to the 14 synapses, so do the multiplication. We've got a lot of bits there. But now it raises a conundrum because I told you they have the same pre and postsynaptic history. Yeah, that's true on the input and the output, but in between there is probabilistic release. Now, something I haven't told you, but I am now, is that on any given axonal input, the probability of release of a single vesicle ranges from 0.1 to 0.5, depending on the strength of the synapse. So the small ones that you saw were 0.1 to 0.2. And that means that for every 10 stimuli that comes flying by, you're at most going to get 1 or 2 vesicles released. It means a tremendous amount of stochastic variability on the input side. How could you possibly store 5 bits of information if you've got this really noisy stochastic release? That we know has been there, for 30 years. Chuck Stevens was the first one who actually did careful measurements. So there is one way for that to be done, and the simplest way would just be to average over releases, right? So if you average, if it's noisy, right? If you keep averaging by the law of large numbers, the average goes down by 1 over, the variance goes down by 1 over n, right? So the standard deviation by 1 over square root of n. And eventually you'll get it to the point where it's as accurate as we see it. And I won't go through the numbers. It's a binomial model where you have n action potentials with a probability of release p. And the question is, what's the CV? Here it is in terms of function of n and p. And then you can then solve that for n, the number of action potentials you need in order to achieve a CV of this here, which is small. So this is going to be a large number. And it also is going to depend on p. And so these two numbers for the small ones they're both small. P is small, it's like a tenth. CV is small, right? So it's going to require a lot of action potentials. And if you just plug in some different p's and different the ends that then you get out from the CV, right? This is the input, this is the output from this equation. You can see that you're going to have to average over many minutes. And even in the most favorable cases at the highest rate with the highest probability, you still have to do over many seconds. So we have to have some machinery somewhere that is an integrator, is capable of integrating over minutes. And we're to look for that. And fortunately, we were collaborating with Mary Kennedy. So Mary Kennedy studies CAM kinase 2. This is an amazing molecule. It's a huge molecule. It's a dodecamer. So as you can see, these subunits, this is one of the subunits, are arranged in two rings that are sandwiched on top of each other. And each of these subunits has binding sites for calmodulin that becomes active when the calmodulin, 4-calcium binds to the calmodulin, the calmodulin then binds to the CAM kinase 2, and that can change the conformation of these side groups here. And that's true for each one of these subunits. And in addition, this causes phosphorylation to occur, which then again changes the kinase activity. And now, at a certain critical point, it self-phosphorylates. And so it goes into a state where it's highly active and stays active. So it's like a switch. Very interesting molecule. And nobody really understands what it's doing there. But our hypothesis now, because if you look at the rates of transition between these different phosphorylation states, it turns out to be in the seconds to minutes range. So we think that the CAM kinase is sitting there averaging calcium as it comes in, which is a surrogate for the release, and using that average over long periods of time to make decisions about whether the synapse should increase in strength or decrease in strength. So that gives us a really strong hypothesis now, and it can be gone in and we can test it. It is known that if you knock out CAM kinase 2, or if you disable it, you block the LTP, so it's on that line. Okay, so let's just see where we've been. So what I've told you is that the spine is a very important compartment, that we now know that the function depends on a spatial scale of nanometers. The postsynaptic density of exactly where the ion channels are located is really important, and the receptors, where the calcium comes in. If it's far away, then it changes the calcium dynamics and the accumulation. It depends on where you are relative to the mouth of the NMDA receptor. If you're right at the mouth, you're going to get activated. The calmodulin will get activated. But if you're just a little bit in, just 10 nanometers in, the calcium binding proteins come and they sop up the calcium, and there's nothing that gets into the interior. Very few molecules. So this is an extremely non-homogeneous, non-well mixed system. And you're not going to be able to average and expect to get the right answer. And then that's why we went to this extreme. This is kind of Full Metal Jacket here. But the beauty is that now we put the big investment into making the model, we can ask questions that would be very difficult to get at experimentally, having to do with the history and some of these very different time scales that lead eventually to LTP, which is where we're headed. Now, this last thing I told you about is something that is really, truly amazing, that we could put a number on the precision with which synaptic plasticity that we think that the neuron is capable of storing that information. Now, something happened recently that you probably have read about in the press because it's really gotten a lot of publicity. It's called deep learning. So what is deep learning? Well, it turns out deep learning is the neural networks that I used to work on back in the 80s, that's why I came to Snowbird, on steroids. So back in the 80s, we had these little networks with one layer of inputs between the inputs and outputs. We call them hidden units. Well, now there's typically 12, which is what you see in the cortex, in the visual hierarchy. But now, why stop at 12? They've gone up to 200, right? But here's what they've discovered, and it's really, I think, relevant for understanding the biology. Here's what they discovered. It's really interesting. So it turns out that you need, in order to train it up, they have a learning algorithm called backprop, which is not biological, but we assume that there's something in biology which can do as well. But you need to have an accurate estimate of the synaptic strength, on the order of 5 bits, in order to be able to train it up, because it's the small changes that are providing you with the learning signal that trains up each layer to be sensitive to the features of the input that are relevant for the discrimination at the output. However, after you've trained up the network, you can then reduce the accuracy of the weights by simply 1 or 2 bits and still get similar performance. And why? Because you have a lot of fan-in, typically thousands of inputs to a single unit. And it's okay if they're stochastic as long as the actual weights that you're picking your coin toss from are accurate. And those weights, the strengths of the weights with the low accuracy produce equivalent behavior. And that may explain why nature has used this very peculiar way of throwing away the accuracy of your synapse by only releasing one set out of every 10 or 5 or 10 times, right? That seems crazy. How could you build a reliable system with a synapse that is unreliable, right? That doesn't make any sense. You're throwing away all of that information that you have stored. And now we know from deep learning that, in fact, you can get perfectly adequate performance, good performance, almost as good performance with stochastic synapses and relatively low accuracy. And the beauty is that you vastly reduce the computation you need to do. Because if you don't release, you don't have to do a multiply, which is what you do in an artificial network. You don't have to resupply the vesicle. In a biological network, you save a lot of energy. So this is really good news. And we saw it didn't require a single simulation. All we had to do was to look at the experiment that nature did for us. Nature created these pairs of synapses so that we can actually look into the accuracy with which nature is capable. And now we actually, I think, are making progress because we can begin to compare what we see in the cortex with what we see in artificial networks, which are reaching levels of performance that are approaching those of humans for speech recognition, for object recognition and images, even for natural language tasks like picture captioning and translation. If you've tried Google Translate recently, it's reached a point where it's really much, much better. It's just within the last couple of months. Okay, well, I want to thank my collaborators. And this is Kristen Harris, who did the reconstructions with us, and she's been a wonderful partner because she's had so much experience with all the different anatomical features of the dendrites. Mary Kennedy, the biochemist at Caltech. So this is Tom Bartol, who's responsible for MCell, and Kaylee Brommer. I didn't tell you what she's doing, but she, in addition to helping with the CA project I told you about, she's also repeating that with the dentate gyrus, looking at these granule cells and seeing the degree to which we can store, how many bits of information can we store in the dentate. So that's her thesis. So thank you all for your attention.
We have time for a few questions. Terry, I'll let you.
Okay, in the back. Well, very good question. And I think the answer, we don't know the answer. And the reason is that for experimental reasons, almost all the work has been done on the large spines. And it is possible that, in fact, the mechanism is... No, we can, but we will. So we have the calcium signal. We know it's different. We know that it's much bigger in the small spine. And that what we don't know yet is the impact downstream. We're doing that right now with calmodulin. But there's this another way in which the synaptic plasticity rule can differ, and we know it's different depending on where you are in the dendrite. So if you're looking proximally, it's symmetric, the LTP and LTD, but if you're up higher, the wing for LTD, the window is much broader. It's like, instead of 20 milliseconds, it's on the order of 50 milliseconds. And it's also known that it's more complicated than I've shown you, that it really depends not just on 2 spikes, but actually on 3. And so there's a lot more we know. In fact, that was very important for us to be able to incorporate some of that into our modeling. Okay, well, I'm glad you asked that question because I'm one of the faculty and a member of a P41. You have one here, Chris here at SCI, and there's one at Pittsburgh, Pittsburgh Supercomputer Center, University of Pittsburgh, and Salk Institute. And what we have proposed to do in our renewal, which we'll find out within the next few weeks, 5-year renewal, is to be able to vary the actual size. Right now, it's fixed to our reconstruction. But what we want to do is to be able to have the machinery downstream reorganize the cytoskeleton, and we can simulate that too with the actin polymers and so forth, and then be able to automatically vary the strength of the synapse, the size of the synapse. We're working right now on something which is important to understand, which is how do you regulate the number of AMPA receptors in the PSD? And what we didn't know until recently is that there's a tremendous amount of exchange between AMPA receptors in the PSD and the extra PSD receptors that are sitting out there. And really, it's a beautiful story. Daniel Choquet has actually followed single AMPA receptors in the membrane with super resolution, and he actually has shown this. This is amazing. And so we've been putting this into our simulations, and it really has consequences that are really, really interesting. So the electrophysiologists who record from single channels have told us the AMPA receptors desensitize very rapidly. So if you give it a shot of glutamate and then 10 milliseconds later another shot, those AMPA receptors are already down, desensitized. So how could it be when in vivo you measure the EPSP and it doesn't desensitize, right? How could that be? And one possibility is that the exchange is so fast that the desensitized get shuffled out and the new ones get put in before the next glutamate spritz, right? So we didn't believe that. We thought that was ridiculous. How could that happen within 50 milliseconds, right? That seems like it's asking a lot. So we put it into the simulation, and it works beautifully. So that exchange process is quite possibly what's really going on. So this is, again, every time you ask a question, right, the answers you get back are often counterintuitive because we can't really think. We don't have a lot of experience with living at that scale, the nanoscale. And so we're gradually building it up by seeing these experiments and trying to understand the actual mechanisms at that level. Another question here.
So one area would be really explore is the maybe that potential very small tumor. I wonder if you model the idea that potential might not get all the way to the very small tumor,
Absolutely right. And we have studied it, but not with M-cell. We have studied it in the context of multiple sclerosis, where you often have failure. But you raise another interesting thing. Again, do not trust anything you read in the textbook, I'm telling you. Okay, if you go to the textbook, what you see is the canonical synapse in the cortex is you have a dendrite here, you have an axon here, and there's a bouton, right? A little button at the end, literally the end of the axon. You never see that in the cortex. It's all en passant. You saw that, right? The axon goes by, and there isn't any ending. The vesicles are kind of massed together at the side of the axon, and there's a big, it's a lot of machinery there, all the snare complexes, and it has to be recycled, and it's an incredibly complex system. But the reality is that you probably will never get a failure in an en passant, because that's where they're swelling, actually. We have the bigger axon. But nonetheless, we know there are conditions like multiple sclerosis where you do get failures. And there's even some evidence in, I think, a spinal cord where this may actually happen under natural conditions, where branches, where axons branch, that's another failure point. And it could actually, with some spike trains, will go through and distribute, but other spike trains may only go through the main one and not through the branch. So these are other possibilities that we can explore ultimately with modeling and then make some predictions. Okay, take Wood here. Right. Exactly. So this is a perfect example of how your intuition, based on just looking at the gross morphology, is misleading. So when you saw the reconstruction, it looks like spaghetti, doesn't it? All of, massive intertwined axons and dendrites and so forth. And what we have found every time we looked is that it masks that apparent complexity, masks this level of precision that nobody anticipated. And I'll just give one example. So, I just told you that the release is probabilistic, and so, gee, that's really noisy, right? So that may actually be a feature rather than a bug. In other words, nature may have actually gone out of its way to make it probabilistic. And we know that it doesn't have to be, because it turns out the neuromuscular junction is almost completely deterministic. When you need to have a high precision, nature can make it, and it decided not to make it in the cortex, right? So I think that we're just beginning to understand the level of precision, and this 5 bits is a good example of something no one anticipated, and it illustrates Leslie Orgul's second law. So Leslie Orgul was a chemist, worked on origin of life, and his second law states that nature is more clever than Leslie Orgul. And he was an incredibly smart guy, one of the smartest I've ever met. And I think it really tells us that we need humility here. We need to step back and say, well, gee, we really just beginning to appreciate how nature managed to put together such an incredibly, detailed machine at the nano level here. We're talking about, over many orders of magnitude. That works reliably most of the time, right? And so let me just end with that, saying that I think we're beginning to sort of, with the right tools and techniques, you need to have those tools and techniques. Otherwise, at the same level of precision as the actual device itself, before you even have a chance of being able to decipher what its actual function might be.
All right, let's thank Terry for a really interesting talk. Thanks.